Good day, everyone. My name is Jay Sung, and I'm an assistant professor at the Mayo Clinic in Rochester, Minnesota. Today, I'll talk about how the gut microbiome is linked to improvement in symptoms of rheumatoid arthritis. Before I begin, I'd like to mention that the work I'll be presenting today was published last month in Genome Medicine. This work was led by a postdoc in my group, Dr. Vinod Gupta. So if you'd like further details on our work, which I was unable to fit in within a 10-minute time span, please see Vinod's paper out in print. Let me begin with a quick introduction on rheumatoid arthritis, or RA. RA is an autoimmune and chronic inflammatory disease which mainly attacks joint linings and causes painful swelling that eventually results in bone erosion and joint deformity. So almost needless to say, RA is a really painful disease in which patients have serious difficulties in performing daily routine activities. There is yet no cure for RA. Fortunately, there are drug treatments that lead to temporary remission of symptoms or reduction in disease activity, such as the one shown here. However, the problem is, it turns out that the patients show tremendous variability in their response to treatment. Some turn out fine after a first dose of a particular drug, but some take longer to respond, often requiring 2 or 3 rounds of doses, while a large proportion do not respond at all, making them bounce from one drug to another until they find something that works. Now, before I begin my main story, I want to briefly talk about how clinical response to a drug is actually measured. There are many ways to quantitatively measure disease activity, such as the Clinical Disease Activity Index, or CDAI for short. CDAI is a mathematical formula that considers the count of tender and swollen joints and also takes into account pain estimates provided by the patient and the physician provider. Minimum CDAI is 0, which means complete remission, and maximum is 76, which means you're in intolerable pain. Depending on the CDAI score, patients can be stratified into 4 disease activity groups: high, moderate, or low disease activity, and also remission. Now with CDAI, you can estimate clinical improvement if one drops in disease activity. For example, when someone goes from high to low disease activity or moderate disease activity to remission. To this point, there's something called the minimum clinically important improvement, or MCII, which is basically a yes or no, depending on whether the patient shows the required minimum drop in CDAI score to be clinically deemed as showing improvement. So for example, if you see on the figure on the right, if a patient is showing high disease activity, then he or she needs to have a drop of at least 12 CDAI points to be considered, at least in the clinic, of showing improvement. What we wanted to do in this study can be described in 2 aims: to identify differences in baseline gut microbiomes between patients who show clinical improvement or not, independent of their previous clinical course. So in other words, regardless of prior treatments, which we can control for in statistical models, is the gut microbiome different between those who clinically improve versus those who do not? And second, can we use baseline gut microbiome data with machine learning tools to predict future clinical improvement? Here's how we recruited patients. We recruited 32 patients with RA at various levels of disease activity. At baseline, we recorded their CDAI score, which again is a quantitative measure of disease activity. We asked them to come back for a follow-up visit 6 to 12 months later. At the follow-up visit, we reported their CDAI score again, allowing us to then determine whether those patients showed improvement or not. For those who show clinical improvement, we call those folks the MCI positive group, which means that they are positive for obtaining the minimum clinically important improvement. There were 12 patients in that group. For those who did not show clinical improvement, we call those folks the MCI negative group, which means that they are negative for minimum clinically important improvement. There were 20 patients in that group. This is a table showing that the 2 patient groups, MCI positive and MCI negative, did not show any differences in sex, age, or race. Also, I'm not showing the data here, but these 2 groups did not show any differences in the type of drugs they were taking. Now, importantly, we're also obtained stool specimens from all 32 patients in both patient groups at both time points. On these 64 samples, we performed shotgun metagenomic sequencing, which allowed us to profile taxonomies and the functional genes. This is a principal coordinate analysis plot showing a projection of all 64 samples. Blue are the microbiomes of the MCI positive group, red are the microbiomes of the MCI negative group, and the 2 samples from the same patient are connected to each other. We found that there was a subtle but statistically significant difference in the samples between both groups. More specifically, when we do a per-MANOVA analysis to see what clinical or demographic features is associated with the variance in gut microbial community composition, we found that the outcome of clinical improvement, which is a yes or a no to showing MCII, is the second highest associated factor with the gut microbiome. Besides MCII, age, use of conventional synthetic disease-modifying anti-rheumatic drugs, or CSD-MARDs, sex, and smoking status also were associated with the gut microbiome. Now at baseline, we find that the gut microbiomes of those who eventually went on to show clinical improvement in RA symptoms had higher alpha diversity and species richness compared to those who did not eventually show clinical improvement. We also found a number of taxonomic ranks and biochemical functions that were differentially abundant at baseline between the two patient groups. Here, I'm just pointing out one in particular, the genus Coprococcus, which was found to be higher in the MCI positive group than in the MCI negative group. Interestingly, Coprococcus is a producer of butyrate, which has been widely reported of showing anti-inflammatory effects. Okay, so what? And where do we go from here? Our next question was, how can we apply gut microbiome information to forecast whether a patient will show clinical improvement? So to this end, we trained a deep learning neural network model for predicting clinical improvement in RA. Specifically, the inputs of our model were all microbiome data, clinical data, and demographic data of all the patients at baseline. This came out to be 448 predictor variables. The output of our model is basically one response variable, yes or no to the question, at follow-up visit, did the patients show clinical improvement in RA? And we tested the performance of our model using leave one out cross-validation. This is a confusion matrix where the rows are showing the actual observed class labels and the columns are for the predicted group labels. Now, as you can see in the first and third quadrants, 16 of the 20 patients who did not show clinical improvement were correctly predicted using baseline features. And all 12 patients who showed clinical improvement were correctly predicted as showing baseline features. Accuracies were 80% and 100%. So the balanced accuracy was an encouraging 90%. Now, going one step beyond classification, we can look under the hood of this machine learning model to see what features were the most informative to the neural network model in correctly making its classification decisions. I won't go into the details of how this is actually calculated, but all I can say for now is that we took all 448 predictor variables and rank ordered them depending on their importance to the model's classification process. As you can see in the upper left, only microbiome features compose the most informative features to classification. The top 5 features were genes for a sucrose degradation pathway, a Parabacteroides species, the Rosebaria genus, and genes for fatty acid metabolism and biotin biosynthesis. In fact, the highest ranked non-microbiome features were the use of CSD-MARDs and patient sex, which were ranked way below as 78th and 87th, respectively. So the main message here is that the gut microbiome is much more important in predicting clinical improvement than clinical and demographic features combined. In summary, gut microbiome is associated with clinical improvement in RA disease activity, higher microbiome alpha diversity and richness in MCI positive RA patients at baseline were found. We also found higher abundance of Coprococcus, a butyrate producer, in MCI positive group at baseline, and microbiome features were more informative in forecasting MCII than clinical and demographic data. So in the long term, we expect our study to contribute towards smart applications of the gut microbiome in the clinical practice. Lastly, I'd like to acknowledge my group at Mayo Clinic, our collaborators, and funding sources for the study. In case anyone has questions on our work, please feel free to contact me directly or refer to our paper online. With that, I'd like to conclude my talk, take any questions or comments from the audience. Thank you for your attention.